20 years, and it wasn't until August of 2022 that we acquired our own field research sites. We've been really building our capacity for field work. I'm super proud of it, and you'll see several photos of this over the next few minutes. For those of you that are not familiar, the Danforth Center is a private, nonprofit, independent plant research institution in St. Louis, whose mission is to improve the human condition through plant science. And my work there comes as part of my faculty role at St. Louis University. I'm in a joint appointment, so I serve both institutions, education and research. All right, so I want to just start out super broad and ask everyone in the audience to just take a moment, bear with me here, and let's think for a minute about maybe think back to when we were kids, or you were a kid, because I don't think I knew anyone in this room when we were all kids. And if you can imagine where you might have been as maybe a 10 or 11-year-old, maybe it's in your parents' kitchen, maybe it's in a classroom somewhere, maybe it's at a playground. And when you were that kid, what did you want to do? What did you want to be when you grew up? Let's remember for just a second. So what comes to mind for me is a baseball stadium. On a summer day, a wind blowing out, players on the field right before the game gets started. They're throwing the ball around the horn. They're taking batting practice. There's vendors rolling up and down the aisles, selling hot dogs, cold drinks. Everyone in the stands is excited and they're ready and they're eager to see the team roll in with a victory. So when I was a kid, my dream, and this is a true story, was to be on a professional baseball team. And it wasn't just any team. My dream was to play for the Chicago Cubs. All right. That didn't happen. That didn't happen, but I am on a scientific team, actually a number of different scientific teams, and I'll be introducing some of those teams today, including the lab group that I lead at St. Louis University. So I'll be talking about the work of many folks on this slide today. I have a great staff of senior research scientists and postdocs shown in the top left corner here. Matthew Rubin and Molly Hanlon, you'll hear quite a bit about them today. Stella Woolchin, who's a proud University of Minnesota alum, and Zach Harris. This is our postdoctoral crew. In the bottom left, there are five PhD students and a master's student in the lab. Dava, Kenneth, Mia, Danielle, Tyler, and then Viva. And then we have a number of St. Louis University undergraduates and technicians who've contributed to our work. Everyone on the team plays an important role in all of our projects. So I'll try to highlight that as we go through. So I didn't make it to baseball, but I did make it to a team. And although my original dream didn't come true, I have other dreams now that I'd like to share with you. And I think some in the audience might share some of these dreams too. So one of the things I'm thinking about now is, can we improve and expand perennial species in our agricultural landscapes? Most perennial crops historically have been perennial woody systems like grapevines. These are some of our most ancient domesticates. And there's some work going on in our groups right now on some perennial work. I have a second dream, and it's a dream that was inspired and that I first came to through my interaction with the Land Institute in Salina, Kansas, through inspiration from Wes Jackson and the breeders who are working on perennial grain crops now. And that dream is, can we develop an agricultural system that mimics nature? We look at a prairie like what we're seeing here at the Shaw Nature Reserve. Most natural ecosystems consist of long-lived perennial species growing together in mixtures. So can we derive and be inspired by and actually model in our agricultural systems those properties which have been so important in ecosystem functioning? So I have a new team and I have a new dream, and that's kind of what I want to talk about today. So the lab group that I run is very broadly focused on perennial plant biology, diversity, and evolution. As I mentioned, we've been studying phenotypic plasticity in clonally propagated grafted grapevines for almost 10 years now. Together with a group of researchers at Cornell and South Dakota State and Penn State, Missouri State, we've been trying to understand how root systems affect traits of shoot systems and how that changes over time and over space. This, unfortunately, I'm not going to be able to talk much about this in this seminar today, but I would love to tell you about it one-on-one or else in other settings today. So if you're interested in woody perennial crops, grafting, phenotypic plasticity, please ask me about it. What I'll spend most of the time talking about in this hour is our work on perennial herbaceous species. So perennial herbaceous species, mostly in the grass and legume families, were really not domesticated. They're just starting to be domesticated now. And I'm not a breeder. I like to describe myself as being breeder adjacent. We're trying to ask questions that are relevant for plant breeding, questions that maybe plant breeders are thinking about, but either don't have the time to or the mandate to answer in their own programs. And these are really basic science questions. So questions like temporal patterns of trait variation and covariation, how trait relationships change over time in perennial systems. We're also trying to use what we learn about plant traits over time in perennial systems and develop ways to expedite and optimize and enhance breeding of these new perennial grains. So the bulk of my talk will focus on our perennial herbaceous work. I'm going to be covering kind of two sections of work here. One focusing on biological patterns of trait variation. So we'll talk about covariation across life stages, across organ systems, and also with spectral signatures. So we're starting to use spectral data in our work as a proxy for biological function. So the first half of the talk will focus on these biological patterns of trait variation. The second half of the talk will focus on the applications of some of these patterns in some breeding applications. So this is me carrying the biomass of a single silphium plant, Silphium integrifolium rosin leaf, a member of the sunflower family that we harvested from our plots last summer. This is not heavy at all. It's like super light. They're really, really bulky. So when we think about what a perennial plant is actually making over the course of a single year, we're trying to understand how flowers relate to fruits, relate to leaves, relate to stems, and relate to roots. All right, and all of this, again, we're asking because we're really motivated by this idea of trying to incorporate perennial species, herbaceous species, grown in some form of mixture, which I get this question a lot. You know, do you think agriculture is going to look like this in the future? No, I mean, really, we're talking about bicultures or polycultures planted in rows that are mechanically harvested, but that are long-lived species growing with more than one taxon in a field. So why are we interested in this? Because through their root systems, plants connect above ground ecosystems, terrestrial ecosystems, to the soil. And for those in the room who are working in root systems, they're so hard to study. You can't see them. You don't know how they're growing. You don't know what's around them. It's just such a conundrum. But plants are so cool because half of their bodies are above ground, and there's a whole other half below the soil that's critical, but really almost impenetrable to science. So these long-lived plants we know they have persistent roots. We know those roots are anchoring soil. We know they're regenerating soil, but we know very little about what that looks like, and we know very little about how what is happening below ground relates to what's happening above ground. So there was a wonderful study published in 2020 by Christine Sprunger, who's a professor at Michigan State University, formerly at Ohio State. I wasn't involved in this work, but I really like to use it to set up what we're doing. This is a study that compared, the title of it is Systems with Greater Perenniality and Crop Diversity Enhance Soil Biological Health. This is a study that compared annual systems, shown in gray, perennials grown in monoculture, and perennials grown in polyculture. So the monoculture's perennials shown in orange, the polyculture perennials shown in yellow. And this just looks at the accumulation of mineralizable carbon on the left and epoxy on the right over an 18-year experiment at the Kellogg Biological Station. And what you can see here is when we look at annual systems, corn and cover crops, corn soy rotations with cover crops, soy corn rotations with cover crops, and compare them to monocultures of switchgrass or miscanthus, shown in orange, or polycultures of poplar, native grasses, early successional communities, or even restored prairies, that the perennial systems have greater mineralizable carbon epoxy in the soil relative to the annual systems, and the perennial systems where there is diversity within them have even greater, these greater metrics of soil health. So we use this to motivate our work as we consider a perennial polyculture agricultural system and the potential impacts that it might have on soil. A lot of people are interested in this work because of not our work, but the broader shift towards perennial polycultures, because we recognize that half of the world's topsoil has been lost in the last 150 years. And we think that perennial systems, specifically perennial polycultures and agriculture, might help stem that. Agriculture, one other statistic, agriculture is the world's largest and most rapidly expanding ecosystem. So the idea here is if we can figure out how to incorporate perenniality and diversity into agriculture, we can perhaps have regional and maybe even global level effects on what's happening with our soil. Of course, the problem that I alluded to earlier is that perennial herbaceous crops were never domesticated. This is something that is being addressed directly here at the University of Minnesota and many other places. Some of you may be aware, even though we had these perennial woody crops domesticated thousands of years ago, herbaceous species were never domesticated. I forgot I threw this in. And the perennial grains are being domesticated today. One of the most exciting recent papers that's come out has highlighted the development of a perennial rice. Has anyone in this room read this paper or familiar with it? Okay, a couple. I like to show this too, because there have been folks who have said, forget it, you can't, there's no way you can develop perennial herbaceous crops. It's too late, we don't have enough time, there's too many trade-offs, there's a million reasons why it won't work. But this paper really demonstrated, it's published in the journal Nature Sustainability, that it is possible to develop a perennial grain crop. This was derived from a cross between Oryza sativa and Oryza longistaminata, a perennial grain. It was planted out in multiple locations in China and compared side by side with annual rice. So in this figure, the perennial rice is shown in red, the annual rice is shown in black. This is a system that was harvested twice per year for 5 years. And you can just see here, the grain yield of the perennial and annual rice was relatively consistent through these 2 harvest cycles for 4 solid years. And then the yield started to diverge in year 5. So for folks who say a perennial grain just isn't going to produce at the same rate as an annual grain, I think this study suggests otherwise. This study also looked at labor inputs, comparing the cost of transplanting annual rice and perennial rice, which are about the same, with the cost of regrowth. Of course, because you have to replant annual rice every year and not perennial rice, labor costs are lower for perennial systems relative to annual systems, as measured in this particular study. That translates into increased economic profit. So again, comparing perennial rice in red and annual rice in black, the output is about the same, but the net profit is greater in the perennial system because of these reduced labor costs. And then lastly, getting back to soil organic carbon accumulation, when we compare our annual systems to the perennial system, soil organic carbon is accumulating at greater rates in perennial systems over time relative to the annual system. So in short, we're inspired. And of course, here in Minnesota and in St. Louis and in many places around the Midwest, folks are looking at an alternative to an annual wheat, an intermediate wheatgrass, Cyanopyrum intermedium. I trust many here are intimately familiar with this great grain trade named Kernza. If you're not, I often get questions about how can I use it, where can I get it? You know, I order it on, well, we mill it from our experiments, or we get it on Amazon from Perennial Pantry. And I bet there's folks in the room who know of other places to get curds of flour. I'd love to hear about it. There's also a fair amount of brewing happening as well. All right, so biological patterns of trait variation. Again, we're trying to lay the biological foundation for the development of perennial grain crops. The work I'm going to talk about now is led by Matthew Rubin, who's a senior research scientist at the Danforth Center. And I want to walk through 3 questions that we've been asking over the last 5 years. First, how do perennial plant traits co-vary across life stages, from seeds to seedlings to year 1, year 2, year 3 growth? Second, how do perennial plant traits co-vary across organ systems? So in this case, I'm talking about systems that are above ground, shoot systems, relative to shoot systems that are below ground, root systems. And I'll talk about some of our recent root work. And then lastly, do spectral data inform our understanding of perennial plant biology, which they do. All right. So we started an experiment in 2019 with 13 different perennial plant species from 3 plant families: the sunflower family, the grass family, and the legume family. Some of these species are species of agricultural importance or interest, like Cyanopyrum intermedium, Kernza, Hordeum brachyanthrum, a barley relative, Silphium integrifolium. Others are things that we just thought are kind of interesting, like Canadian wild rye, Elements canadensis. So we had a broad diversity of species from 3 plant families. We planted over 20,000 seeds from multiple accessions from each of these species. And we did this over the course of 2 years. In the first year, cohort 1, we had a relatively small number of accessions with about 48 seeds per accession, 246 accessions total. In the second year, we planted a second cohort where we increased the number of accessions, but decreased the number of seeds per accession. And this was the starting point of this experiment, again, which is geared at looking at how traits change over the lifespan of a diversity of plants. So we planted seeds and seedlings in 2019 and 2020. Before we did that, we scanned and weighed every seed that we planted. We monitored germination rate and proportion, and we harvested a subset of samples so we could measure above and below ground biomass in seedlings. We also collected spectral data using a machine called the Crop Reporter, as well as near-infrared and RGB images on an automated phenotyper at the Danforth Center that we call the Lemnitech. Once all of this was done, these individual plants were taken out to the field where we've been monitoring in-field traits over the course of 3 years. And I say monitoring, actually, we monitored, we dug them all up at the end of last year. So this is what the automated phenotyper looks like at the Danforth Center. This is a growth chamber that holds 1,140 plants. These plants are maintained in controlled conditions. They're all barcoded, and they move around on this little go-kart or runway type of system where they're automatically watered and weighed every day. And then they're imaged from the top down and from the side in RGB and in near infrared. So this is a way that we comprehensively capture early life stage growth rate. I'm very proud to say that we doubled the number of species that had ever been on this machine with our first experiment, although some were very poorly behaved and grew too quickly and fell off, took their partners off with them, but we got it going. So once we collected all of those data on seeds and seedlings, we loaded up the U-Haul and trucked them out to the Shaw Nature Reserve, where we had built a field site. This is a field site that is within a 10-acre deer fenced enclosure that we later built a small rodent fence. We landscape fabricated it, and then we co-opted children to help us plant this experiment. So this is what the plot looked like. Those are my kids, so I think it's fine. Although that kid is huge now. That was like so long ago. Anyway, this is what the experiment looked like. It's about 1/3 of an acre. It had over 2,000 plants. We didn't plant all of the 20,000 seeds that we attempted to germinate. And this is where we had been monitoring traits over time. So again, in the mature plantings over the course of 3 years, what we were really looking at was overwinter survival. Did the plant make it? Flowering time, height, and then yield. And so how do perennial plant traits co-vary across life stages? This is a correlation matrix that summarizes all of the information that we have for all of the species and all of the individuals. So this is a very broad, rough assessment. And the first thing that you'll notice, each one of these squares, if it has a color, it is statistically significant. If that color is in the blue family, it is a positive correlation. If the color is a shade of red, it is a negative correlation. So the first thing we saw is that there are very strong patterns of both positive and negative correlations across species. Some other things we noticed is that seed weight and seed area are negatively correlated with germination proportion, but not germination time. Seed weight and seed area, if we look all the way across this, are also related to the traits that we've been measuring in the field: overwinter survival, flowering time, height, yield, and the proportion of viable seed. Now, these specific relationships vary on a species by species basis, and again, this is basically a summary, but we are in the process of going species by species now. We can also look at germination proportion and germination time, and we see that those traits are also associated with traits we see in the field, including height, yield, and the proportion of viable seed. So when we look across time at covariation among traits, we do see a range of different and very interesting relationships. So I'm going to just take a moment to talk about one of our favorite perennial herbaceous species, Silphium integrifolium, or rosinweed. This correlation matrix is similar to the one that I just showed, but again, it's only for our Silphium accession. This looks a little bit, you'll notice a little more white space, but still significant, some significant correlation. So if we look at seeds, so the weight, the area, the length, and the full width, we can just see off the top here that there is a negative correlation between these seed image traits and the prevalence of orange rust, the height, the head number in year 1 that we measured, and the head number in year 2. So there is this relationship, and I should have mentioned, this is not new. People have been looking at relationships between seeds and seedlings and mature plants for a long time. I think what is new here is that we're applying some newer phenotyping approaches and hopefully more automated phenotyping approaches to capture this information. We also saw a relationship between growth rate and stem rust and total seeds in year 1 and year 2, and then the flowering time. The problem with that study that I just mentioned is that it really only looked again at this above ground part of the plant. And when we think about perennial species and why we're interested in them for future agricultural systems, one of the things we're interested in is what is going on below ground. So we can see these relationships above ground, which are interesting and which, as we'll see in a minute, have some applications we hope in breeding. But the real action is below ground. So Matthew led that project. He initiated it in 2019. A couple of years ago, we recruited Molly Hanlon to come along and help us with root biology. Molly is a senior research scientist now at the Danforth Center who did her PhD working in a corn root lab. These two, whenever I see them together, it makes me so nervous. But this is the two of them sitting in one of the offices at the Danforth Center, and nobody's moving. Okay, these are Home Depot boxes full of biomass that we had run out of space, and there was this very narrow. I mean, you would think we are all hoarders, and maybe we are, right? Maybe we are. The very narrow space, and these two squirrel up in the back corner and talk data. And this is how I found them a couple weeks ago. So Molly came in to try to add the below ground component to this giant study. So what we tried to do was to first extract soil cores from a subset of individuals at this experiment at the Shaw Nature Reserve, and then phenotype roots through those cores. We then went through and excavated via shovelomics all of these plants, washed them off, well, transported them to a place where we could actually do the root washing, wash them off, and then have been phenotyping these roots. So again, these are the year 3 root systems that we were able to excavate with a shovel. So it's really root crowns that we're talking about. Sadly, we are not getting into the deeper parts of the root system, although we're trying that in another approach now. So what Molly has shown, and again, we're looking at these multiple different species, wild perennial species that have some interest for potential crop development. Elamis, Canadensis, glaucus, and virginicus, these are Canadian wild rye and relatives. Helianthus maximiliani, a sunflower relative. Silphium integrifolia, we've already seen that one, sunflower relative. Steinopyrum intermedium, intermediate wheatgrass or kernza, and then Trifolium ambiguum. This is a cura clover that some folks are interested in as a potential perennial cover crop. Okay, so in this figure, the top bar shows the height of the plant. The bottom section shows the depth of the root. So we see here that plant height and root depth distributions vary with species. Of course, there is species-specific variation both above and below ground. Across all species, total root length shown on the left and deep root length shown in the middle don't correlate directly with plant height. However, the proportion of deep roots indicated with D95 correlates with plant height. So deeper, we're seeing that taller plants have deeper roots, and we saw this across these different species. Then when we went to intermediate wheatgrass, kernza, for destructive root sampling, shovelomics, so destructive root sampling, coring, and then tried to look at the relationships between root data obtained in those two ways and above ground trait measurements, we're starting to see some interesting patterns related to resource allocation and trait covariation. So these graphs here show different aspects of the above ground and below ground traits and how they relate to one another. I'll just highlight a few. We can see here, again, this is in kernza, that rhizome weight is positively correlated with shoot weight, and root weight is also positively correlated with shoot weight. This is of interest because there are some who have argued that there are trade-offs between below ground and above ground structures, and that may be true for some systems, maybe true for annual systems. Maybe it was a one-off, I don't know. But the data that we're starting to see here suggests that we really don't see that trade-off between, for example, yield and rhizome weight or other root traits. Again, looking at intermediate wheatgrass, the deep root length, deeper than 50 centimeters, we can see that deep root length positively correlates with increased shoot biomass and yield. So here, the third line across looks at shoot biomass. The fourth line of figures looks at yield. And we can see that deep root length is positively correlated with both shoot biomass and yield. So as Molly says, big deal, bigger plants, bigger roots, but it is kind of important, I think, as we think about how we might select on these plants to drive both yield, but also the below ground ecosystem functions we're so interested in. All right, so the third thing I wanted to talk about here in this section on biological patterns of trait covariation is to talk a little bit about spectral data. So is anyone here using spectral data or reflectance signatures in their work? A couple. Okay, good. I would like to hear about this. So part of the experiments that I described a moment ago involved a fair amount of spectral data. So that included both crop reporter data, which collect information on reflectance patterns associated with some photosynthetic processes, as well as hue, saturation, and value, HSV, and NIR. These are the images coming from the Lemnitech, that's the video that I showed. And at each time a picture was taken, this is one silphium genotype and then a second silphium genotype. We can see they're growing at different rates, but we were interested if the spectral signatures were different between these lines. So this was actually a very rich data set. For the experiment I described, we ultimately put 4,560 individual plants through this system representing these 13 species, 3 families. We had a total of 350 populations or accessions, and they were imaged for an average, not an overage, an average of 10 days over the course of 3 to 5 weeks, yielding a total of 50,000 images. And again, a part of these images were reflectance data that we are trying to correlate with other traits. So just a few things we learned. This is a picture from our greenhouse after the plants came off the Lemnitech. So you can see just by looking, like there is some color differences in leaves. And if you look at plants, as we all do, you know that. But when we look at, for example, hue, and look at hue patterns for the different plant families, again, these are taxonomic families, Asteraceae, Poaceae, Fabaceae, there are differences in the spectral signatures. We can use a machine learning approach, a random forest approach to predictively assign individuals correctly to the right family based on these data. We can also do this at the species level. So this figure shows hue, saturation, and value. These are color metrics, reflectance patterns. Each color bar here is a different species. And we can again use this machine learning approach and models to correctly assign individuals to the right species. The 2-letter abbreviations are species based on their spectral signatures. We took it a step further and looked at plant genetic families, and we were also able to show relationships between spectral signatures and plant genetic families. So this is all going to become important in just one moment. The spectral data that we observed, which are kind of highlighted here, also show not only do they differ by taxonomy and species and family, but they also correlate with traits that we later measured in the field. So they're positive for most and negative correlations for some between spectral signatures measured on seeds and seedlings with overwinter survival, flowering time, height yield, and the proportion of viable seed traits measured later in the field. Okay, and we saw the same thing when we looked at just a single species in silphium. Okay, so I want to just wrap this up and spend the last 10 minutes or so talking about what all of this means and how we're using it in breeding. So the first part of this talk was focused on biological patterns, both temporal patterns and others, in how traits of perennial plants change and relate to one another. So what we've shown is that traits that are expressed in the earliest life stages in seeds and seedlings strongly covary with traits expressed in mature plants in the field. And we've seen this across multiple species and multiple families. Above ground traits, including biomass and yield, are positively correlated with below ground biomass. And then spectral data that we recorded in early life stages first varies by taxon, but also seems to covary with agronomically important traits observed later on in the field. Okay, I love this picture of Jack and Isabella. All of this work is a ton of time in the field. It's hard, heavy labor, and folks are doing a great job. I try to get to the field 1 day per week, but I don't always make it. All right, so the last part of the talk here is going to focus on breeding applications. And what I want to do is show how we're building on this foundation of basic knowledge, basic understanding of the biology of perennial herbaceous species to try to expedite and optimize and enhance breeding of these wild species for perennial polyculture systems. All right, so I showed before, again, that there are strong positive and negative correlations between spectral traits observed early in the lifespan with traits observed later in the field. So we're using this we're using that base of knowledge to fuel a phenomic prediction, phenomic selection study. So plant phenomics encompasses multidimensional data that describe some aspect of plant biology. Phenomic selection then uses what we call high dimensional secondary traits. So this is things like near infrared or other non-destructive absorbance measures. Some people are using hyperspectral imaging, multispectral imaging. Often these absorbance measures are taken on seeds and seedlings in early life stages and used to generate phenomic estimated breeding values, which are then used in a predictive framework. So we are trying this approach out in 3 species, which I've mentioned at least 2 of them already: intermediate wheatgrass, silphium or silkflower or rosinweed, and sainfoin. Our vision here is to see if we can reimagine how crop domestication happens in this era of high throughput phenomics. Can elite individuals of perennial species be predicted by phenomic relatedness models that are based on seed and seedling high dimensional traits? And then the second thing we're looking at is whether phenomic selection can enhance or even replace genomic selection to accelerate breeding. Now, I want to just say one thing about this second point, and then I'm going to not talk about it again. With these new species that are just kind of coming into the attention of breeders or other biologists like me, often we don't have the basic genomic resources, a sequenced genome, a high throughput genotyping pipeline to apply genomic selection. It's not far off. We're not that far off, right? I mean, it wouldn't take that long to do it. Might not even take that much money. But we're interested in seeing if phenomic selection can be kind of like the step 1 of a process that helps enable the diversity of species that are entering into these domestication pipelines. And we're hoping that it's cheap. I mean, we're hoping that we can do it easily and cheaply. And we're trying to capture that financial information as well. So I just want to be clear, I'm all in favor of genomic selection. I'm just not sure it's the right approach for species that are at this very, very early stage of selection. I'll just note, I think many of you are very familiar, there's an active and super productive genomic selection program in intermediate wheatgrass right now, which is yielding incredible results. There's also a silphium integrifolium genotyping foundation that's being built at the moment. So again, we're not far off, but it's just a matter of what can we do right now. Okay, so for the breeding applications of this talk, I'm going to focus on the question, can elite individuals of perennial species be predicted by phenomic relatedness models that are generated based on seed and seedling high dimensional traits? This is another phenomenal team that I have the good fortune to be a part of. The work is again led by Matthew Rubin in collaboration with David Van Tassel and Eric Cassetta, Catherine Turner at the Land Institute, Brandon Schlotman, here's David again, and Lee Behan. We have a number of folks, some of you might know Sydney, who is out in our field she's been there a couple times. Shannon and Emily, who are helping us collect these here as well. This project is kind of a complicated one, and I just want to introduce the experimental design here in some boxes. So the way this is working is for each of these 3 species, sainfoin, silphium, and intermediate wheatgrass, we obtained a bunch of seeds. We then germinated them and applied a drought stress as seedlings. So we're also asking whether stress affects this whole process. So there were seedlings that were droughted and seedlings that were watered for each of the 3 species. Then we planted those plants out into the field in Missouri and also in Kansas. So we have these seedlings that were droughted and not out in 2 locations right now. Okay, and we did that over the course of 2 years. So we had a cohort go out in 2021 and a cohort go out in 2022, and they're in the ground now. So before things made it to the ground, again, we individually scanned, and this is definitely not a high throughput method, but we are working on this. We individually scanned and tracked every single seed. So every individual has a label. And then we collected high dimensional traits, literally hundreds of features. So features would be like a wavelength collected using a handheld spectrometer or the phenotyper. We collected high dimensional traits for seeds on the top row and seedlings for the species that are shown above. So we've had Kernza, Saintfoin, and Silphium one round through. The second round of Kernza and Saintfoin is done, and the third is going in the fall. So these are just the different types of spectral data: hue, saturation, and value, daily, near infrared. And we collected these using different handheld spectrometers called link squares. There's a few others, Innospec, SIO, Cuber. We've actually been trying out different handheld spectrometers to see. They cover slightly different wavelengths, and they also have strengths and weaknesses. So our favorites are now InspectorView, Innospec, and the SIO. The other ones, for various reasons, we're no longer using. So this is, again, a very rich data set collected on seeds and seedlings. Then all those seeds and seedlings were put out into the field, as I described, in Missouri and in Kansas. And we've been collecting data on overwinter survival, plant size, flowering time / maturity, as well as yield traits for each one of these species. So again, we're trying to see if we can predict, based on these spectral signatures, elite individuals with desirable traits at maturity. So I want to show you what we've done so far in one species, Silphium, for one trait, ligule number. And this is we're just kind of learning how to do this. So this is where we are at the moment. It's underway for the other two species, but I don't have anything I can show today. So phenomic prediction models for ligule number in Silphium. This is what a beautiful Silphium inflorescence looks like. Like any Asteraceae, it has ray florets and disc florets. The ray or petal on the outer flowers is called the ligule. And in Silphium, this is an important point. In Silphium, the ray florets, or the florets that have ligules, are the ones where seeds are made. So it's important. That's an important fact, because what we're hoping is if we can predict ligule number, it may be a proxy for seed production. Okay, so we generated relationship matrices based on these high dimensional seed and seedling spectral data on all the seed Silphium individuals phenotyped to date. When I say we, I mean Matthew Rubin evaluated more than 100 combinations of high dimensional seed and seedling data sets. So each spectrometer, each set of features is a data set, and we're testing what combinations of which data sets produce which models. We train those models for ligule number using Silphium planting training sets in 2 sites. So the 2 sites were in St. Louis and in Kansas. We were unable, I know, I always have to put this in. We were unable to collect data on the 3rd training set because it got mowed. Science, it happens. Okay, that's okay, we're moving on. So we use then those models that are based on those 2 training sets to predict ligule number in a novel environment, a test set that was in Kansas, that we call a Kansas 3 test set. And we predicted ligule number based on the relationships that were established in the training set. And again, these relationships are derived from different combinations of spectral data taken on seeds and seedlings. Okay, so what did we find? This is a preliminary model that shows the correlation between ligule number predicted based on the relationship matrices that I mentioned and the numbers of ligules that were observed in the actual test set. And these are 3 different relationship matrices that are different combinations of spectral data sets. So the correlations range from roughly 0.2 to 0.45, which is significantly greater than random. So this indicates that we can accurately predict ligule number based on relationship matrices that are derived from these high dimensional data that were taken on seeds and seedlings. And this level of correlation is on par with what we see in other phenomic prediction studies and other genomic prediction results. Okay, so then what we did based on the predictions is we crossed individuals in the lowest one and the top and bottom 1% and 5% as far as ligule number goes. So these figures just shows what an individual that has a lot of ligules looks like and what an individual that has not a lot of ligules looks like. So crosses were made, and next year what we'll do is grow the seed from those crosses and compare the ligule number between the 4 crossing groups to a random set of open pollinated seed. Okay, one last thing, and I think this is like my last data slide. Following making those predictions in the novel Kansas environment, we actually went and counted ligule number on all the plants in the Kansas 3 site, and then tested for the correlation between the predicted values derived from the training set and the observed values that we actually saw for the ligule number. And we observed a similar correlation, about 0.35. So this is important because it shows that individuals in that Kansas site, that novel site, which are unique genotypes in a unique environment, that for this trait, the phenomic prediction model is robust. So it's robust, as I've written here, to genotypic and environmental variation. So this demonstrates that we can actually make some meaningful predictions about a trait. Actually, I have one more slide. A particular trait using spectral signatures. All right, last slide. This figure shows the correlations between predicted and observed values using different combinations of spectral data. I showed you ligule counts, and we talked about the correlations that we came in around 0.25 to 0.45. We've also looked at other traits like blotch, seed weight, total head number, orange rust, total seed stem count, total seed weight, and viral score. And we're seeing a range. So one of the things we're doing now is evaluating how we're collecting data in the field and which models might be robust for these different traits. Okay, so to wrap up this section on breeding applications of patterns of trait covariation, we showed, I think we're starting to show that elite individuals with traits of interest for perennial species can be predicted based on spectral data taken on seeds and seedlings. We showed it for silphium, and we're working on it for sainfoin and for intermediate wheatgrass. So I started out by sharing this dream. Can we improve and expand perennial species in our agricultural landscape? I mentioned we're working on grapes, didn't talk about it, but that's rolling ahead as well. I think based on the data that we showed, there's maybe some new ways that we can try to expand the species that we're working with. And then can we develop an agricultural system that mimics nature and perenniality and diversity? We didn't really get to this, right? We only got to the as Wes Jackson called the hardware part, like the actual perennial grain crops, which we're beginning to try to develop and to try to support the development of. But so can we actually go from a prairie vision to a practical farm? I hope so. This is the picture I started with, and this is another experiment I didn't get a chance to share. But in this experiment, what we've got is intermediate wheatgrass, Cyanopyrum intermedium, growing in monoculture, and then in biculture with alfalfa. And we're looking at how the traits that I've talked about today vary based on monocultures with fertilizer treatment, and a biculture. And we're also looking at how those relationships, for example, above and below ground trait covariation shift based on site. This is an experiment that's replicated in 7 locations from Western Kansas, Central Kansas to Vermont. So the next step here is to think about how we can use this foundation to move towards a polyculture system. Maybe it looks like this, maybe it looks something different, that mimics nature. Okay, I think that's it. Thank you so much for listening. I'm happy to take any questions. Again, it's a giant team effort. These are the funding sources that supported the work I talked about today. I'm happy to take any questions. Yes,
That was a really great talk. I've got kind of a longish question about temporality.
bring it.
There's been a lot of studies that have looked at the temporality and variance partitioning in modern single season traits like maize plant height. They've seen that the effect or the percent variance explained by genotype, environment, and G by E kind of vary throughout the growing season.
Mhm.
Do you have any indication of how those sort of big components, G, E, and G by E, how those impact development across multiple seasons? Is environment more important in any particular year?
It's a really great question. I have the same question. I don't know the answer, but I am into it, and I think that's where we need to go next. So we do have some mapping populations in the ground now that will allow us to look at the genetic architecture, the genetic component of some of the traits that we're interested in, and the genetic architecture of those traits. They are perennial plants, so we can look at that over multiple years. They are planted in multiple sites, so we can try to get at that. We're thinking about it, and I don't know. I'll also just share, we have another study with another mapping population that in grapes, actually, that's planted across 5, maybe it's 4 sites now, trying to get at the same thing. So the genotype by environment partitioning of temporal variation in traits. If you know of work that has done that, I would love to know. But that's, yes, I hope we can get there someday. Yeah, great question. Thank you. Yes.
Show these strong correlations between the spectra and some of these traits within species. Have you looked at within related individuals, how those correlations compare to the within species correlations?
Yeah, we have, and I took that slide out, but there are significant relationships between spectral signatures. Their spectral data significantly correlate with genetic family, not plant taxonomic family, but genetic family across many wavelengths. So I hope, I'm not sure if this is the question you're answering, but we are seeing that relationship as well. If that's not the right answer, do you want to ask again?
No, I think yeah, that was basically what I was asking. So I guess also to just follow up, and how do those compare to just like the more broad within species correlations?
I think, yeah, so good question. I haven't explicitly quantified that, so I'm going to tell you my eyeball answer.
Okay.
I think that the differences in spectral signatures increase as the genetic divergence increases. So if you're looking at the level of genetic families within a species, we are picking up differences in spectral signatures, but they are not as dramatic as the differences we see when we look from one species to the next, which are not as dramatic as the differences we see when we look at grasses versus legumes. Yes.
So you have a lot of this data. Have you thought of doing any metabolic traits and the metabolite, how those vary with other biological traits, or how other micronutrients used for human health vary within these as well?
Yeah, that's another really great question. We have looked at this in grapevines, and we have quantified basically ion concentration. So from aluminum to zinc, 20 different ions. And we did this in an experiment where we held the shoot system constant and we manipulated the root system because we wanted to know if different root systems were taking up different concentrations of ions that we were measuring in this common clonally propagated grafted scion. And what we learned is that It's really fascinating. So ion concentrations vary. First, they vary by rootstock. So different rootstocks are taking up different ions. Second, although that is a very subtle effect. Second, they vary by the position of the leaf on the vine. So younger leaves have concentrations of ions that are different than older leaves. And in some cases, it's higher. In some ions, it's higher in the younger leaf. Some ions, it's lower, higher in the older leaf. Those patterns change over the course of a season. So if you look at spring versus raisin versus harvest in a grape situation, those patterns shift. They also change from year to year. So it's a hugely dynamic, as you would imagine, process. And you might be thinking, well, who cares about ions in grapevine leaves? What about the berries? Well, I even have that same question. And that is actually a proposal we're developing right now. Yeah, good question. Yes.
Allison, you didn't mention this, but it seems to me that with perennial crops, another aspect that could be a real focus is how much carbon is being sequestered. You know, because putting roots, I don't know, 2 or 3 feet down,
Mhm.
that's carbon that's come out of the atmosphere that's not going back in for a long time.
Yeah.
Is that something that you guys have asked? Because that seems to be a very big difference between these perennial and annual systems.
It's a great question, and I got that exact same question just last week, and we are starting to ask that. And it's kind of interesting, it's obvious that that's a focus, but it's, people are so, this is a major focus that we need to be addressing. So we are starting to figure out how we can quantify carbon over time. Like one of the challenges we're having with roots, again, because we can't see them, is, you know, what happens to them? Like, do they, is it a root system that is stable over the course of multiple years? Or is it a root system where parts of it are dying back, just as we see grapevine, you know, shoots die back in the winter? We're trying to learn that, and also then try to quantify that aspect of carbon capture, carbon sequestration. And we're not there yet, but we're thinking about it.
But in the pictures of the intermediate wheatgrass, the thing that's pretty obvious, right, is if you remove the above ground plant, there's still a whole lot of stuff left in the soil.
Right.
And that's not going anywhere for a long time. So,
Yeah. So
so
how do we quantify that, what that is?
yeah, it seems like a hard problem, but quite an important one in terms of understanding the.
So we are working with mini rhizotrons and some of these experiments that are, you know, tubes in the soil that allow us to capture images of roots in real time. And we're also thinking about spectral systems that might be able to tell us some aspect of soil chemistry. But this is a non-working system at the moment. It is an idea. If anyone in this room is doing that kind of stuff, I'd love to hear about it. But we don't have it going quite yet. Yeah, it's a great question. Yes.
When you see a correlation between like stem rust and seedling traits, do you think that's physiology or linkage?
Or something else. I mean, I don't know. I think that's in that system in Silphium with rust, which there's multiple rusts, and they seem to blink on and off, and then they show up in a really massive and destructive way. You might have, the short answer is I don't know the answer to your question. But our ability to predict rust or viruses in Silphium based on seedling spectral data was very bad, very low, which is extremely disappointing because it's a major interest of ours. So I'm not sure. And I think we don't know really the genetics of resistance to those. I don't, at least, maybe someone here does, of resistance to those particular pathogens. So I'm not sure, but somebody, maybe you, should take a good look at this. Yeah. Guys, these are some really nice questions. Anything else? Well, as you know, my dream is to just do our little part to try to learn what we need to learn to maybe move the needle even the teeniest bit in this direction. So any thoughts you have, I'd love to hear them either today or via email another time. Thank you so much for coming. Appreciate it.
will be here in this room for a little while, like I think half hour to be able to chat and have some conversations
Oh, good.
for first meeting, so
Okay,
feel free to talk more.
thank you, Erin. I did not know that.
Surprise!